Chairman Dr. Jason Khan from Department of Food and Biotechnology, Faculty of Agriculture and Veterinary Science, Jyoti Vidyapeeth University, Jaipur. Today I am going to discuss about the topic fermentation. This topic belongs to the fundamental of microbiology subject. So fermentation, what is fermentation? Fermentation is a metabolic process. We know that microorganisms have the capacity to utilize some uh, carbohydrate source and convert them into the alcoholic beverage. So what is that? This is the this is their metabolic activity by which they convert some molecules into another forms. So fermentation is a metabolic process that converts sugar into acids, gases, or alcohol. So the sugar is being converted into the acids, gases, and alcohol by the microorganism. And microorganisms have some particular enzymes which convert these organic molecules into different types. And it occurs in yeast, in bacteria, and also in the oxygen starving muscle cells. And in as in the case of the lactic acid fermentation. So lactic acid is also produced in lactic acid fermentation method. And fermentation chemical is a chemical process by which molecules such as glucose are broken down anaerobically. Fermentation is a forming that occurs during the manufacture of wine and beer, and it is a process of at least 1,000 years old. The 14 results from the evolution of carbon dioxide gas. So this was not recognized until the 17th century. French, French chemist and microbiologist Louis Pasteur in the 19th century used the term fermentation in medicines to describe the changes brought about by yeast and microorganisms in the absence of the air. So, in the absence of the air means they are growing anaerobically, and they also recognize that ethyl alcohol and carbon dioxide are not the only products of the fermentation. Means microorganisms are producing more and more productions by the process of the fermentation. Okay. If we talk about the definition, fermentation is the chemical transformation of organic substances into simpler compounds by the action of enzymes, complex organic catalysts, which are produced by the microorganisms such as the molds, yeast, and bacteria. Enzyme act by hydrolysis, a process of breaking down or pre-digesting complex organic molecule to form smaller molecules into simpler nutrients. So sugar, yeast will utilize sugar, and they after utilizing they will go for the fermentation process, and they will produce alcohol and CO2. What is the history of the fermentation? Fermentation is a natural process. People apply fermentation to make products such as wine, uh, they made cheese and beer, okay, in very longer before time. Like this pasture became the first to use it. The first solid evidence of the living nature of yeast appeared between the 1837 and 1838 when three publications appeared by the C. Kinnear, D. Tower, and T. Swan and T. Cuisine. They concluded as a result of the microscopic investigation of the yeast was a living organism that reproduced by budding. The word yeast should be noted traces its origin back to Sanskrit word meaning boiling, and it was perhaps because wine, beer, and bread were each basic food in Europe. But most of the early studies on fermentation were done on yeast, with which they were made. After that, bacteria uh, were also discovered. The term was first used by the English in the late 1840s, but it did not come into general until 1870, and then largely it was used in combination with the germ theory of the disease. What are different types of the respiration? Aerobic and anaerobic respiration. These are the two types: aerobic respiration and the anaerobic respiration, and they both are fermentation based. Now talk about the aerobic fermentation. Aerobic fermentation means that the oxygen is present. Okay. Wine, beer, and acetic acid or vinegar such as apple cider vinegar need oxygen in the primary of first stage of the fermentation. When creating acidic vinegar, for example, exposing the surface of vinegar to as much of the different another products. When creating acetic acid, for example, exposing the surface of the vinegar, oxygen is required. It is much more required, and it creates a healthy flavored vinegar that correct pH. That fermentation is also type. Anaerobic fermentation is a method cells used to extract energy from carbohydrates when energy or other electron acceptors are not available. That's about it. And this differentiates it from anaerobic respiration, which doesn't use oxygen but does use electron accepting molecules. Okay. So it comes outside the cells, and the process can follow glycolysis as the next step in the breakdown of the glucose and the other sugars to produce molecules of adenosine triphosphate that create an energy source from the cell. This method of cell is able to generate nicotinamide, adenine, dinucleotide from the reduced form of the nicotinamide, adenine, dinucleotide, and NADH. Okay, is a molecule which is necessary to continue glycolysis, anaerobic fermentation, relies on enzymes to aid phosphate group. Okay, they need a phosphate group to an individual adenosine diphosphate molecule to produce ATP, which means it is a form of substrate level phosphorylation. And this contrasts with the oxidative phosphorylation, which uses energy from an established proton gradient to produce ATP. There are two major types of anaerobic fermentation: ethanol fermentation and lactic acid fermentation. Okay. Uh, though both of these store NADP plus to allow a cell to continue generating ATP through glycolysis. And the second one is the ethanol fermentation. Ethanol fermentation converts two pyruvate molecules, the product of glycolysis, to two molecules of the ethanol and two molecules of carbon dioxide. The reaction is a two-step process in which pyruvate is converted to acetaldehyde and the carbon dioxide first by the enzyme pyruvate decarboxylase. Yeast and certain bacteria perform ethanol fermentation. They perform ethanol fermentation where pyruvate from glucose metabolism is broken down into ethanol and carbon dioxide. And the net chemical equation for the production of ethanol is C2 C6H12 O6 means glucose. Okay. C6H12O6 is converted into the H5OH. Two molecules of C2H5OH are produced, means it is ethanol, and two molecules of the carbon dioxide are produced. Okay. Ethanol fermentation is used for the production of beer, wine, and wheat. And it's worth noting that the fermentation in the presence of high level of active results in the production of small amount of ethanol, which is toxic and it is consumed. Lactic acid fermentation is also a type of fermentation. Lactic acid fermentation is a biological process by which glucose and other six carbon sugars, also disaccharide of six carbon, are, are also like a glucose, sucrose, lactose are converted into the cellular energy and metabolic lactic. Okay, so lactic acid is produced, lactate is being converted. Okay, the pyruvate molecules from glucose metabolism may be fermented into the lactic acid. Lactic acid fermentation is used to convert lactose into lactic acid in yogurt production, and it also occurs in animal muscles when the tissue require energy at a faster rate then the oxygen can be supplied. Okay. So what is the reaction? C6H12 O6 will be converted into the lactic acid. Okay. CH3CHOOH. Okay. The production of lactic acid from the lactose and water may be said that C12H12O11 means lactose. Okay. C12H12 S22 and O11 with water hydrolysis and convert it into the lactic acid. Yogurt is made by fermenting milk. It's a highly rich protein rich milk product, and it is rich in the protein, calcium, and probiotic. Okay, different type of fermentation can be said that homolactic fermentation. The fermentation is in homolactic and heterolactic, two type of fermentation. So first, homolactic fermentation, the fermentation in which only the lactic acid is produced. There is no any side product produced after reaction is known as the homolactic fermentation. And heterolactic fermentation, the fermentation in which the lactic acid is produced along with some another products like gases. So that is known as the hetero fermentation. Now talk about the mechanism of the fermentation. Fermentation takes place when the electron transfer chain is un unusable. And it turns NADH and pyruvate produced in glycolysis into NAD+ and organic molecule. In the presence of the oxygen, NADH and pyruvate are used to generate ATP in respiration. And this is called the oxidative phosphorylation, and it generates much more ATP than glycolysis alone. For that reason, that cells generally benefit from the avoiding fermentation when oxygen is available. So in the first step, glycolysis is common to all fermentation pathways. Okay. So first C6H12O6 means glucose plus 2 NAD plus 2 ADP and 2 pyrophosphate will convert into the pyruvate. Okay, so that is the basic. Now pyruvate pyrophosphate is inorganic phosphate and 2 ADP molecule and 2 pyrophosphate are converted to 2 ATP and 2 water molecules are produced. In oxidative phosphorylation, the energy for ATP formation is derived from an electrochemically proton gradient generated across the inner microbial membrane. And the glycolysis has substrate level phosphorylation at the point of the reaction, and humans have used fermentation to produce food and beverages since a longer time. For example, fermentation is used for the preservation in the process that produce lactic acid as found in the sour foods, pickles, cucumber, kimchi, yogurt. So we know these products are being utilized for a longer time, as well as they are also used. People are using this fermentation for producing alcoholic beverages like wine, beer. So these all are the examples. What are the different products from the fermentation? Wine, beer, lactic acid, vinegar, yogurts, cheese, sauerkraut, kimchi, pepperoni. So these all are the products of the fermentation. So and these products can be produced in the industry level. And what are the advantages and disadvantages of fermentation? So advantage of fermentation are that the lactic acid can be produced and it can be produced for the ATP production. And disadvantage, if we talk about the disadvantage is that the production, that fermentation are that production can be slow and the product is impure and need further treatment after the production. So this is the disadvantage. So I hope that the students have better understood all things about the fermentation. In the next video, I will discuss about the aerobic and anaerobic respiration. So my lecture is over here. If you have any query, you can ask me. So thank you very much.